If you want to understand why cancer rates in industrialized nations have increased 43% over the last 30 years, despite billions spent on chemotherapy research and early detection campaigns, don't look at genetic mutations. Don't look at environmental carcinogens. Look at what's happening inside your large intestine right now, where trillions of bacteria are either producing compounds that starve cancer cells of fuel or creating an internal chemical environment where malignant cells thrive unchecked and undetected. Here's a number that should stop you cold. Right now, inside every single person watching this, microscopic clusters of abnormal cells are forming. Your immune system identifies and eliminates thousands of potentially cancerous cells every single day. This is not theoretical. This is measured biological reality. The question is not whether precancerous cells form. The question is whether your body's surveillance and elimination systems function efficiently enough to destroy them before they establish blood supply, evade immune detection, and grow into tumors that require surgical or pharmaceutical intervention. I'm an emergency medicine physician and oncology researcher. And after nearly 15 years in emergency medicine and 3 years deep inside the literature on cancer metabolism and the gut microbiome cancer axis, I've come to understand something that fundamentally contradicts the genetic determinism most people have been taught about cancer. Your cancer risk is not written in your DNA. It is written in the metabolic products produced by the bacteria living in your digestive tract. And one specific bacterial species, when present in adequate populations, produces a short-chain fatty acid that has been demonstrated in over 200 peer-reviewed studies to selectively induce apoptosis in cancer cells while leaving healthy cells completely untouched. Most people believe cancer is primarily genetic. You inherit bad genes, or you accumulate mutations through aging or environmental exposure, and eventually those mutations cause cells to turn cancerous. And while genetic mutations do play a role, that model is fundamentally incomplete. It cannot explain why genetically identical twins have dramatically different cancer outcomes. It cannot explain why cancer rates vary by more than tenfold between populations with similar genetic backgrounds but different dietary patterns. And it cannot explain the single most important fact about cancer metabolism that has emerged from the last 2 decades of research. Cancer cells have a metabolic Achilles heel. They are obligate glucose fermenters. Unlike healthy cells, which can flexibly burn glucose, fatty acids, or ketones for energy depending on availability, cancer cells are metabolically inflexible. They depend almost entirely on glucose fermentation for ATP production, even in the presence of oxygen. This is called the Warburg effect, discovered nearly a century ago, but only recently understood in terms of therapeutic targeting. Think of healthy cells as hybrid vehicles that can run on gasoline, electricity, or diesel, depending on what fuel is available. Cancer cells are like old single-fuel engines that can only run on one specific type of gasoline. If you restrict that fuel while simultaneously introducing compounds that poison their specific metabolic pathway, you create an environment where cancer cells cannot survive while healthy cells continue functioning normally. This is selective metabolic targeting. And it does not require pharmaceutical intervention. It requires understanding which foods feed your gut bacteria that produce the compounds cancer cells cannot tolerate. Now, here's what most oncologists won't tell you, not because they're hiding it, but because it falls outside their training and practice scope. Approximately 70 to 80% of your immune system resides in your gut-associated lymphoid tissue. The bacterial composition of your microbiome directly regulates whether that immune system can identify and destroy cancer cells or whether it remains blind to malignant transformation. Specific bacterial species produce metabolites that train your T cells to recognize cancer antigens. Other bacterial species produce compounds that suppress immune surveillance, allowing cancer cells to proliferate undetected. The difference between a microbiome that protects you from cancer and one that enables it comes down to the presence or absence of specific bacterial populations. And the single most important species in this equation, the one that produces the compound with the most robust anti-cancer evidence across multiple cancer types, is a bacteria that most people have never heard of, but that can be dramatically increased through consumption of one specific category of food that costs less than $5 per week. Before I explain the science, I want to be very clear about what I am and am not claiming. This is not a cure for diagnosed cancer. If you have active malignancy, you need oncological care. Chemotherapy, radiation, and surgery save lives. But what I'm about to explain represents something that conventional oncology does not address: the creation of an internal metabolic environment that is fundamentally hostile to cancer cell survival and growth. This is cancer prevention and metabolic terrain optimization. It works alongside, not instead of, medical treatment. The bacterial species is Faecalibacterium prausnitzii. It is one of the most abundant bacteria in healthy human colons, typically representing 5 to 15% of total gut bacteria. And the compound it produces is butyrate, a 4-carbon short-chain fatty acid that functions as the primary fuel source for your colonocytes, the cells lining your large intestine, while simultaneously acting as a histone deacetylase inhibitor that triggers apoptosis in cancer cells. Now let me walk you through exactly what happens at the cellular level when Faecalibacterium prausnitzii populations are abundant and producing therapeutic levels of butyrate, and why this matters for preventing and potentially reversing early-stage malignant transformation. Mechanism 1. Selective cancer cell apoptosis through histone deacetylase inhibition. Inside every cell in your body, your DNA is wrapped around protein complexes called histones. How tightly that DNA is wrapped determines which genes can be expressed and which remain silenced. Enzymes called histone deacetylases, or HDACs, remove acetyl groups from histones, causing DNA to wrap more tightly and genes to be turned off. Cancer cells exploit HDAC activity to silence tumor suppressor genes, the genetic off switches that would normally trigger cell death when DNA damage is detected. Butyrate is a natural HDAC inhibitor. When butyrate enters a cell, it blocks HDAC enzymes from removing acetyl groups. This keeps DNA loosely wrapped and allows tumor suppressor genes to remain active. In healthy cells with normal DNA, this has minimal effect because those cells are already properly regulated. But in cancer cells where tumor suppressors have been epigenetically silenced through excessive HDAC activity, butyrate reactivates those suppressor genes. The cancer cell suddenly recognizes that its DNA is damaged beyond repair, and it initiates programmed cell death. Think of cancer cells as criminals who have bribed the judges to ignore their crimes. Butyrate is the internal affairs investigation that removes the corrupt judges and reinstates the legitimate legal system. Once proper oversight is restored, the criminals are immediately convicted and sentenced. The cancer cell's own machinery executes it. Researchers at the German Cancer Research Center in Heidelberg conducted experiments using human colorectal cancer cell lines and measured the effects of butyrate at concentrations achievable through dietary fiber fermentation in the colon. At butyrate concentrations of 2 to 5 millimolar, which represents the range produced by healthy microbiomes fermenting adequate dietary fiber, colorectal cancer cells demonstrated apoptosis rates of 63 to 78% within 48 hours. Normal colonocytes exposed to identical butyrate concentrations showed apoptosis rates below 5%. The selectivity was remarkable. The compound was killing cancer cells at rates 12 to 15 times higher than healthy cells. What this means for you is that maintaining adequate populations of butyrate-producing bacteria creates a continuous anti-cancer surveillance system in your colon, where any cells that undergo malignant transformation are rapidly identified and eliminated before they can establish tumors. Mechanism 2. Metabolic starvation through glucose uptake inhibition. Cancer cells require approximately 18 times more glucose than normal cells to produce equivalent ATP energy. This is the metabolic consequence of the Warburg effect. They ferment glucose inefficiently and must consume massive quantities to meet energy demands. Cutting off glucose supply to cancer cells is therefore a logical therapeutic strategy. But it's extraordinarily difficult to achieve through dietary glucose restriction alone, because your liver continuously produces glucose through gluconeogenesis even when you're fasting. Butyrate provides a different approach. It doesn't reduce glucose availability. It blocks cancer cells' ability to import glucose from the bloodstream. Cancer cells express abnormally high levels of glucose transporter proteins, particularly GLUT1, on their membranes. These transporters are the doors through which glucose enters cells. Butyrate downregulates GLUT1 expression specifically in cancer cells while leaving glucose transport intact in normal cells. When cancer cells lose GLUT1 expression, They cannot import sufficient glucose even when blood glucose levels are normal or elevated. They experience what researchers call metabolic catastrophe. Their energy production collapses. They cannot maintain membrane potentials. They cannot replicate DNA, and they die from energy starvation while surrounding healthy cells continue functioning normally. A study published in Cancer Research in 2021 from researchers at Johns Hopkins University examined the effects of butyrate on pancreatic cancer cells, one of the most aggressive and treatment-resistant cancer types. They found that butyrate treatment reduced GLUT1 expression in pancreatic cancer cells by 64% within 72 hours. When they measured glucose uptake using radioactively labeled glucose, cancer cells treated with butyrate showed 71% reduction in glucose import compared to untreated controls. The cancer cells were sitting in a glucose-rich environment, but could not access the fuel they required for survival. Think of this like disabling the fuel pump on a car. The gas tank might be full, but if the engine cannot draw fuel from the tank, the vehicle stops functioning. Butyrate disables the fuel pump specifically in cancer cells while leaving normal cells' fuel systems intact. Mechanism 3. Enhanced immune surveillance through regulatory T-cell differentiation. Your immune system's ability to detect and destroy cancer cells depends on proper training and regulation of T lymphocytes. Naive T cells must differentiate into either effector T cells that attack foreign cells or regulatory T cells that prevent autoimmune reactions. The balance between these populations determines whether your immune system can identify cancer cells as threats without attacking your own healthy tissue. Butyrate directly promotes regulatory T cell differentiation in your gut-associated lymphoid tissue. These regulatory T cells, or TREGs, migrate throughout your body and fine-tune immune responses. Paradoxically, increasing TREGs actually improves cancer surveillance rather than suppressing it, because properly regulated T cells can distinguish between self and non-self more accurately. They attack cancer cells while sparing healthy tissue. Additionally, butyrate enhances the cytotoxic activity of CD8-positive T cells, the specialized immune cells that directly kill cancer cells. Butyrate increases their production of interferon gamma and perforin, the molecular weapons they use to destroy target cells. Researchers at the University of California, San Francisco tracked immune cell populations in 87 adults with varying levels of butyrate-producing bacteria confirmed through metagenomic stool sequencing. Participants with high Faecalibacterium prausnitzi populations, defined as greater than 8% of total bacterial abundance, showed regulatory T cell counts 41% higher than participants with low F, prausnitzi below 2%. More importantly, when researchers measured CD8-positive T cell activity using cytokine production assays, the high F prausnitzi group showed 37% higher interferon gamma production, indicating more potent anti-cancer immune activity. This demonstrates that the bacterial composition of your gut directly programs your immune system's cancer surveillance capacity. You are not stuck with the immune function you were born with. You can enhance it through deliberate microbiome modification. Mechanism 4. Reduction of genotoxic secondary bile acids. Your liver produces primary bile acids to emulsify dietary fats, enabling absorption. These primary bile acids travel to your colon, where gut bacteria convert them into secondary bile acids through a process called dehydroxylation. Some secondary bile acids, particularly deoxycholic acid and lithocholic acid, are genotoxic. They directly damage DNA, promote inflammation, and drive colon cancer development. Populations of pathogenic bacteria, including Clostridium and Bacteroides species, specialize in producing these harmful secondary bile acids. When these bacteria dominate your microbiome, your colon is continuously bathed in genotoxic compounds that cause the DNA mutations that initiate cancer. Faecalibacterium prausnitzi and other butyrate-producing bacteria compete with these pathogenic species for resources and territory. When butyrate producers are abundant, they suppress pathogenic bacteria through competitive exclusion and through production of antimicrobial compounds. This reduces the production of genotoxic secondary bile acids, lowering the DNA damage rate in your colonocytes. A prospective cohort study from the Netherlands followed over 4,200 adults for an average of 11 years, tracking both microbiome composition through periodic stool sampling and colorectal cancer incidence. Participants with high baseline Faecalibacterium prausnitzii abundance had a 62% lower risk of developing colorectal cancer over the follow-up period, compared to participants with low F. prausnitzii. When researchers measured fecal bile acid composition, the high F. prausnitzii group had 49% lower deoxycholic acid concentrations, indicating reduced genotoxic exposure. Think of your colon as contested territory. Pathogenic bacteria are constantly trying to establish dominance and produce compounds that damage your DNA. Beneficial butyrate-producing bacteria are the defensive force that keeps pathogenic populations suppressed and your colonocytes protected. When the defenders are strong and numerous, cancer cannot gain a foothold. Now here is the exact protocol for dramatically increasing Faecalibacterium prausnitzii populations and butyrate production based on what the microbiome research tells us works. Faecalibacterium prausnitzii is an obligate anaerobe. It cannot be consumed as a probiotic supplement because it dies immediately upon oxygen exposure. You cannot eat it. You must feed the populations already living in your colon. And the specific substrate that F. prausnitzii ferments most efficiently is a category of fiber called resistant starch. Resistant starch is carbohydrate that resists digestion in your small intestine and reaches your colon intact, where bacteria ferment it into short-chain fatty acids, including butyrate. There are 4 types of resistant starch, but type 2 and type 3 are most effective for butyrate production. The single most concentrated source of type 2 resistant starch is raw, unmodified potato starch. This is not potato flour. It is pure starch extracted from potatoes and sold as a white powder. Bob's Red Mill potato starch is widely available and costs approximately $7 for 24 ounces. Each tablespoon contains approximately 8 grams of resistant starch. The therapeutic dose, based on research showing significant microbiome shifts, is 20 to 30 grams of resistant starch daily. This translates to 2.5 to 4 tablespoons of raw potato starch. You must consume it raw. Heating resistant starch above 140 degrees Fahrenheit converts it to regular digestible starch, eliminating its prebiotic properties. Start with 1 tablespoon daily for the first week. Mix it into cold water, unsweetened almond milk, or a protein smoothie. Stir vigorously because it does not dissolve easily. Consume it in the evening, ideally 2 to 3 hours before bed. Why evening? Because colonic fermentation peaks during sleep, when intestinal motility slows, allowing bacteria maximum time to ferment the resistant starch. Increase to 2 tablespoons in week 2 and 3 tablespoons by week 3 if you tolerate it well. Some people experience gas and bloating during the adaptation period. This is normal. It indicates active fermentation. Symptoms typically resolve by week 4 as your microbiome adjusts. Type 3 resistant starch comes from cooked and cooled starchy foods. When you cook potatoes, rice, or beans and then refrigerate them for at least 12 hours, some of the starch molecules crystallize into a form that resists digestion. This retrograded starch functions similarly to raw potato starch. Practical sources include cold cooked potatoes eaten as potato salad, cold cooked white rice eaten as rice salad, and cooked and cooled navy beans or lentils. Aim for 1 cup of cold cooked starchy food daily in addition to your raw potato starch dose. Here are the clinical pearls that amplify results. First, you must consume adequate total fiber. Resistant starch alone is insufficient. Faecalibacterium prausnitzii and other beneficial bacteria also require fermentable fiber from vegetables, fruits, and whole grains. Aim for 40 to 50 grams of total dietary fiber daily. This means large portions of vegetables with every meal. Second, avoid antibiotics unless absolutely medically necessary. A single course of broad-spectrum antibiotics can reduce Faecalibacterium prausnitzii populations by 70 to 90%, and recovery can take 3 to 6 months. If you must take antibiotics, immediately begin high-dose resistant starch consumption during and after the course to accelerate recolonization. Third, minimize artificial sweeteners, particularly saccharin, sucralose, and aspartame. These compounds have been shown in multiple studies to reduce beneficial bacteria, including butyrate producers, while increasing pathogenic populations. If you need sweetness, use small amounts of raw honey or fruit. Fourth, ensure adequate omega-3 fatty acid intake. EPA and DHA from fish oil support the growth of butyrate-producing bacteria. Aim for 1,000 to 2,000 milligrams of combined EPA and DHA daily from wild-caught fatty fish or molecularly distilled fish oil supplements. Fifth, consider adding inulin-rich foods to your diet. Inulin is a prebiotic fiber that synergizes with resistant starch to promote butyrate production. Rich sources include raw garlic, raw onions, raw leeks, Jerusalem artichokes, and chicory root. Consume at least one of these foods daily. Sixth, track your response through stool characteristics. As butyrate production increases, most people notice that stools become more formed, bulkier, and easier to pass. Bowel movement frequency often increases to 1 or 2 per day, if it was previously less frequent. This indicates healthy colonic function and active bacterial fermentation. Now let me tell you the mistakes that sabotage this protocol. Mistake 1 is heating the potato starch. I cannot emphasize this enough. Raw potato starch mixed into hot coffee or cooked into baked goods becomes regular starch and loses all prebiotic properties. It must remain raw and unheated. Mistake 2 is expecting rapid results and discontinuing after 2 weeks. Meaningful microbiome shifts require 6 to 8 weeks minimum. Faecalibacterium prausnitzii populations do not explode overnight. They gradually expand as you consistently provide their preferred substrate. Patience is mandatory. Mistake 3 is consuming resistant starch while continuing to eat a diet dominated by ultra-processed foods, high in emulsifiers. Polysorbate 80 and carboxymethylcellulose directly damage the mucus layer protecting your gut bacteria and reduce beneficial bacterial populations. You cannot build a healthy microbiome while simultaneously poisoning it. Mistake 4 is assuming that all fiber is equally beneficial and eating massive amounts of insoluble wheat bran thinking it will produce similar effects. Wheat bran is poorly fermented by colonic bacteria. It adds bulk but produces minimal butyrate. You need fermentable fibers, specifically resistant starch and inulin, for butyrate production. Mistake 5 is taking broad-spectrum probiotic supplements with 15 different bacterial strains, thinking more diversity is better. Most commercial probiotics do not contain butyrate-producing species. They contain lactobacillus and bifidobacterium, which are beneficial but do not produce butyrate. You are spending $40 per month on supplements that do not address the specific bacterial deficiency we are targeting. Mistake 6 is consuming resistant starch inconsistently, having it 5 days 1 week and then skipping the next week entirely. Bacterial populations respond to consistent substrate availability. Sporadic feeding produces minimal population expansion. This must be daily for 12 to 16 weeks minimum. Let's lock in everything we covered. Cancer is not primarily a genetic disease of bad luck and inherited mutations. It is a metabolic disease of cellular environments that either support immune surveillance and apoptosis of damaged cells or enable malignant transformation and tumor growth. The single most important determinant of which environment you create is the metabolic products produced by bacteria living in your colon. Faecalibacterium prausnitzii, when present in adequate populations above 8% of total gut bacteria, produces butyrate at concentrations that selectively induce apoptosis in cancer cells through histone deacetylase inhibition, starve cancer cells by blocking glucose uptake, enhance immune surveillance through regulatory T-cell differentiation, and reduce DNA damage by suppressing production of genotoxic bile acids. You cannot consume F. prausnitzii as a supplement. You must feed the populations already in your system through daily consumption of resistant starch. 20 to 30 grams daily from raw potato starch and cold-cooked starchy foods combined with 40 to 50 grams of total dietary fiber creates the colonic environment where butyrate-producing bacteria thrive. This is not alternative medicine. This is microbiome science published in Cancer Research, Cell Metabolism, and Nature Communications. The intervention works. It is safe. It costs approximately $10 per month, and it addresses a cancer prevention mechanism that conventional oncology completely ignores. Your cancer risk is not predetermined. Your immune surveillance capacity is not fixed. The metabolic environment your cells exist within is not static. These are dynamic systems that respond to the bacterial metabolites being produced in your colon right now. The internal terrain you have reflects the bacteria you've been feeding. The terrain you will have 16 weeks from now reflects the choices you make starting today. 2 tablespoons of raw potato starch costs approximately 35 cents. 1 cup of cold-cooked potatoes costs less than 50 cents. You do not need expensive pharmaceutical intervention. You do not need boutique probiotic formulations. You need to feed the specific bacterial populations that produce the compound cancer cells cannot tolerate. I need you to do 2 things. First, if this information was valuable, please share this video with someone you care about who has family history of cancer or who is currently fighting cancer. This could give them a metabolic advantage that conventional treatment alone cannot provide. Second, make sure you're subscribed to this channel. I see messages from people who have watched 20 of my videos and never realized they weren't subscribed. It's free, it takes 2 seconds, and it ensures you don't miss content like this. In my next video, I'm covering the single food that most people eat 3 to 4 times per week, thinking it's healthy that is directly feeding pathogenic gut bacteria, increasing intestinal permeability, and driving the chronic inflammation that underlies virtually every degenerative disease. The research is clear and almost nobody is connecting these dots.